Hello, welcome to our course Applied Environmental Microbiology. I'm your instructor for this course and my name is Gargi Singh. By training, I'm an environmental engineer in the Department of Civil Engineering, and this is very important to highlight to suggest that environmental engineering was not always called as environmental engineering. When we started this field, where we understood that taking care of environment is also a responsibility of a civil engineer, it was referred to as sanitation engineering, where the interest was just to take the waste material and put it away from our site, or maybe put it in a black box system and ensure that our surface water bodies are clean. However, as science developed further, we understood the biological and chemical mechanisms that allow us to keep our environment clean and sustainable. Still, when it came to biology of the environment, very little was known, and it was often assumed that biology works in predictable ways because it has members that act like little black boxes. And I'll give you an example. Some of you might have background in civil engineering or chemical engineering and might be familiar with wastewater treatment. And for those who are not, very briefly, wastewater treatment are processes that use sewage and the domestic wastewater we generate, put microbes in it and allow the microbes to degrade all the biodegradable organics. And then what we are left with is clean water. For many decades, civil engineers and other professionals who worked with sanitation engineering considered the microbiology of wastewater treatment as a group of heterotrophs who simply degrade the organics. But when science developed further, we understood that there's not just one kind of microbe or microorganism that is at work. So what used to be collectively referred to as biomass when we talked about sludge now started being respected in a way and distinguished in sense that now we had heterotrophic bacteria, autotrophic bacteria, we had anoxic and oxic microorganisms, and this is how we started respecting that not all microbes are similar. As the science of microbiology developed even further, we understood that even within these broad classifications such as heterotrophic microbes, there is a huge diversity. There are many different kinds of microbes. In past 3 decades now, microbiology has given us techniques such as genetic sequencing, where we can actually sequence the genome and the genes of bacteria and understand how similar or how far apart they are from each other. And with onset and discovery of these wonderful techniques, what we have now is a much broader and more clear vision of applied environmental microbiology. Unfortunately, in many regions of the world, India included, the microbiology of environment and its applications are not really well understood and are mostly confined to the domain of pure research and is ascribed as something that perhaps a biotechnician would do or somebody with a biological background would do. And this is where I want to give you a small introduction that you do not have to be familiar with biology, let alone microbiology, in order to understand this course really well. What is important in this course from you is that you pay attention to what's taught and covered in the lectures and honestly, sincerely do the assignments and the question answers that I will share with you during this course. Now, today being the first lecture, I would like to take some time to make you more familiar with what we are going to discover together in this course and how it might be applicable for you. Now, in microbiology in general, we talk about microorganisms. We want to know who they are, and this is where microbiology and biology get very tricky and put off many engineers and scientists and science students, because there is a perception in microbiology we need to memorize so many things by rote. And I assure you, in this course, you do not need to memorize different names. You don't need to know how Cranoarchaeota is different from Euarchaeota, but you do need to understand how the microbiology in itself works. And what information do you need in order to understand why microbes are doing what they're doing, and in the first place what they're doing, and how you can tweak that in order to serve your environmental purposes. Which brings us to the second bullet here. The first aspect is to understand what microorganisms are present, how they work, and why they do what they do. And what it results in is an understanding of the immense diversity of microbes and how they evolve from one kind of microorganism to another microorganism. And speaking of diversity of microorganisms, it's really beautiful to note that most microbiologists, including myself, believe that we have barely scratched the surface. We believe that we know less than 1% of microbes that are there on our earth. And the fourth bullet here talks about ecology, which is where do they live and how do they interact with each other. And this is particularly important for environmental engineers, environmental scientists and technicians. We want to understand how microbes behave with each other and how they behave with the environment, how they are influenced by the environment and how do they influence the environment in return. Microbiology in itself has two legs on which it stands. One is the scientific aspect and the other is the application or the engineering aspect. The science wants to know the nature of microbes, how the microbes function, what's the fundamental process of life, what's the biochemistry of life. Whereas the application aspect and the engineering aspect wants to know, okay, how can I use this information that I have to improve our living standard, to benefit our humanity, to make our civilizations more sustainable so that we can live happily on this earth for a longer time. And the application aspect also looks into medicine, engineering and agriculture, and now we have an emerging field of industrial microbiology, and a lot of what you'll study in this course will actually help you address any of these applications. In this course we are not focusing as much on science, and that's why you get a good riddance from all those scientific names and long enumerations of microbes, and you can directly jump into how we can use this information that we have to better our world, to make better medicines, to have cleaner surface water, to have cleaner groundwater, cleaner air now, and just how to be healthier. Now, this is very important to understand that why applied environmental microbiology is very relevant today. We think of Earth in terms of lithosphere. In terms of lithosphere, it has a lot of blue in it, so all the maps, so there's a lot of water and then there is land. What is hiding in this picture are the tons of microbes that actually inhabit the Earth. And if we look at from biomass perspective, the percentage of carbon that's stored in microbes, we can say that the life on Earth is mostly in microbial form, not in the higher forms of life that we see. And let's look at the origin of Earth, and this will cement further our understanding that Earth is a microbial planet. It is said that Earth is nearly 4.6 billion years old, and the life started some 4 billion years ago. That's origin of cellular life. Okay, now we are understanding that non-cellular forms of life started way before 4 billion years, but definitely the cellular life started 4 billion years. That's where the microbes came. And then somewhere between 4 and 3 billion years we got anoxic phototrophic bacteria, and this is very beautiful to understand. At this time, for all these billions of years, Earth was anoxic, which is another way of saying there was no oxygen on Earth. Earth was an anaerobic environment, and humans, as now we are, could not have existed, survived at all on this Earth then. However, a miracle started happening sometime around 3 billion years, when between 3 billion and 2 billion years ago, cyanobacteria, a green beautiful bacteria, beautiful under microscope by the way, green cyanobacteria came over, evolved on Earth. And what this cyanobacteria started doing, it started using the light to produce food, which is something we are familiar with as photosynthesis in the case of plants, and started generating oxygen. So over the course of next billion years, the Earth was slowly oxygenated. So from this primitive form of life, and I'm using primitive in sense of time, not necessarily in sense of complexity, definitely not in sense of complexity, I want to be clear, but in terms of time, the life started also incorporating and exploring the aerobic forms of existence. So around 2 billion years ago, modern eukaryotes, now this is a technical term, eukaryote, prokaryote, eu means true, karyon means nucleus. So something that has a particular kind of cell, which we call as are called as eukaryotes. And it might be helpful to mention that humans are eukaryotes. All the higher order of life have eukaryotic cell, and the uniqueness with eukaryotic cell, as we'll see later, is that they have a nuclear membrane in which they store the genetic material. So some 2 billion years ago, these higher forms of life, the modern eukaryotes, came into picture, and then lo and behold, very soon we had algae. We had this photosynthetic eukaryotic organisms, and then much near the present, some 500 million years ago, shelly invertebrates came, vascular plants, mammals, and humans are relatively a recent phenomena, very recent phenomena. So we can see for nearly 4.6 billion years of Earth's existence, we have at least 3.5 years of only microbial existence. And if we look at in terms of biomass, more organic carbon is stored in microbes than in all higher forms of life combined, and thus we are assured as microbiologists that Earth is a microbial planet. Now in this slide we have 2 beautiful pictures. On left we have a scientist showing off a plate with different microbes growing. Now in this plate, it's very beautiful to note that each of these colonies have emerged from a single bacterial cell. These are bacterial colonies, by the way. And on the right side we have a picture, and this is an artist's impression of three-dimensional picture of microbes of different morphologies, different shapes. So if you look at this visually, on the left in our plate we have microbes that look very similar. The colonies that they make are somewhat circular and start with red, and as they spread out they get more yellow and then even white. On the right side we have these beautiful, strange and very distinct geometries. Some of them are cocos shaped or round shaped, some of them are rod shaped, some of them look like these galaxies, and some of them look like dots that are clustered together. And what this wants to highlight is that on a physiological level there is immense diversity in nature as represented by the right picture, and what we humans have been able to capture in lab is a small fraction, only one kind of microbe. And that's the reasoning behind the popular saying that we have only scratched the surface of environmental microbiology. This is in terms of physiology, so this is how microbes look. Now what microbes are beneath their looks is even more complicated and more interesting. So in higher order of life we see 2 mammals who stand upright on 2 legs with 2 hands, an upright body and a head, and we assume, okay, they're humans, they must be similar to each other, and we do know that genetically we humans are very similar to each other. In physiology we are very similar as well. However, in case of microbes, we cannot have similar ideology doesn't work for microbes. 2 microbes that are perfectly spherical and maybe respond similarly to different light that we throw on them might be very distinct and very unique in their behavior. For example, E. coli, 2 strains of E. coli have exactly similar morphology. One might be benign and might actually help when it is in my gut. The other might kill me in a couple of hours. So same species of microbes that look very similar, have similar morphologies, might be very different in their activity. And let's say even if they're similar in their activity, they might be very, very different in their genetics. So we'll get through what these genes are, what the genetics is, but for now just know that genetic diversity tells me the essential differences that are there in the makeup, in the code of life in these microbes. So in here you have a dendrogram which has been represented in a tree of life format, showing each of these streaks represent a particular kind of microbe, very distinct from each other. The closer 2 microbes are to each other, for example all the blue ones that have clustered here together and then the brown ones have clustered, the closer they are to each other, we can say that more similar these microbes are to each other. The color represents the broad phylum that they belong to, and I can speak with good level of certainty that even though if there are 2 bacteria are clustered closer, for example the yellow or proteobacteria, and even if 2 lines are very close to each other, we might come under the impression that, oh well, they might behave similarly, yeah, but they might be as different as we are from algae. So in this particular picture here, which says genetic diversity, two lines that are very close to each other and have same color might be very different in their behavior and in their genes. Now this slide talks about a very important aspect of essential microbes. Why is this important to know? Why do I want to know about tree of life? Why do I want to know about genetic and physiological diversity? Because there are microbes that directly impact our health, that impact our environment, impact our industries such as agriculture and other industries. And this is on right, there's a picture from, I think, Time magazine. This was the cover of that magazine, showing how human beings are inhabited by microbes. And I think this is a great time to share this, in case you didn't already know, that in our human bodies we are a minority. And what this means is, for every single human cell in your body, we have at least 10 non-human cells, 10 microbial cells. So just take a moment to ponder about this. We are a minority in our own body. Now, are we really humans then? And this is the picture that Time used to illustrate this novel finding that we have. And now we know that microbes affect us in terms of nutrition. They actually will allow us, and we'll see this very soon, that given the right microbes in the gut, in the gut we might never face nutrition deficiency, or if we don't have those good microbes, we might really suffer problem. And coming back to the essential microbe, this picture shows some wonderful research that is going on right now to see how microbes impact our health. Now, it's a very important thing for you to understand that even though we talk about environmental engineering, environment is anything that surrounds us. So we can have at least 3 different kinds of environment. Broadly speaking, we have an external environment and then we have an internal environment. And internal environment would include, for example, my gut microbes. What's going on in my gut? We know that human gut is heavily colonized by bacteria and other microbes. And they are essential. Most of them are very important for our health. If we lose them, we fall sick. Then we have microbes in the outer environment, which impact things like our climate, which impact how much oxygen goes out in the atmosphere. They influence how our waste degrades, how our landfills behave, how our wastewater treatment will be successful or not, what problems we might face, will our sludge float or not. And the latest research, as represented in the slide, is showing that there is no clear division between external and internal environment. The external influences the internal and the internal influences the external. Let me give you an example. You're sitting in a room, clean room like the one I'm standing in right now, and a dirty, muddy dog walks in. Then the dog, because the dog is wet, let's imagine, and when the dog shakes itself to get rid of the water, he also sprays these bioaerosols with a lot of microbes in the air. You happen to inhale some of them. Now these microbes that are in the air, thanks to the dog that walked in, will colonize your nose, your nostrils, maybe your upper respiratory system, your skin definitely, and most probably most of them would be benign and you'll be fine, in fact healthier now because of the immense diversity in your body. But maybe one of them is not good for you and you might fall really sick. So this is external environment impacting internal. Another example would be, I drink water, it has pathogens in it and I fall sick. Now let's look at internal impacting the external. One of the major environmental challenges in front of our country today is antibiotic resistance, and this is a classic example where the internal exchanges antibiotic resistance with the external, and external gifts antibiotic resistance to the internal. So in very soon, and this is also the new motto of our leading research department of biotechnology, that we have to move towards one health. So environmental health, public health and veterinary health all have to come under one umbrella, because we can no longer distinguish between internal and external. However, in this course we'll be focusing mostly on external environment. And this is a beautiful paper that's pretty recent, and we'll talk about this paper later, but for now I think this is enough to know. And this is antibiotic resistance threat, and I wanted to share this, and that how TB, antibiotic resistant tuberculosis, is a big issue in India. And even though it sounds like a very medical problem, well, antibiotic resistant infection, doctors take care of it, medical professionals, medical scientists, biotechnicians, but this is very important. The latest research suggests that the root cause of resistance in tuberculosis or any other pathogen for that matter is not what's happening just inside the hospital, just inside the body and what the drugs are doing to bacteria, but a large role is played by natural environment. And this is where environmental engineering has to jump into action to help combat antibiotic resistance. Alrighty, and this is cyanobacteria, by the way, pictures of cyanobacteria under microscope in lake, and remember, this is the bacteria that generated oxygen for the first time on earth. The other important reason why we should spread awareness of applied environmental microbiology is that microbes not only impact health, whether it's environmental, public or veterinary, but they also impact our climate. They impact how our nutrient cycles move on our earth. For example, here we have nitrogen cycle and carbon cycle. Both are driven by microbes. And this is where it becomes very important to understand that depending on how microbial communities behave, our climate will respond to it. And I would like to quote one of the early heroes of microbiology, Louis Pasteur, that the role of infinitely small in nature is infinitely large, and this is exactly what we hope, this is exactly the message that I hope to pass on to you, my dear students, in this course. So, because the role of infinitely small in nature is infinitely large, let's understand the infinitely small in nature, and we start with the fundamental unit of life, the cell structure. In this slide you will note that there are two different kinds of cells. On the top we have this very simple container with a gray container with a blue inner lining and some liquid inside and this convoluted green mass. Now let me give it more scientific terms. The layer outside is actually cell wall. The blue layer is cytoplasmic membrane. The orangish liquid here with dots in it is cytoplasm, and then this green convoluted mass is actually nucleus, so it's the genetic code. This particular kind of cell is called prokaryote. Pro meaning primitive, karyote means cell. So this is a primitive cell, not in terms of complexity again, but in terms of time when it first evolved. Now it's very important to note that in this cell it has a very simple geometry and it has only one container, so it's like a one room house. In the bottom we have a eukaryotic cell, and I have already mentioned that eu means new, karyon means cell. So in this we have a container, an orange container with a blue inner lining, some orangish liquid here with dots, and look, there are multiple organelles within this compartment. There is this pink mitochondria, there are these blue endoplasmic reticulum, and then there is this green nucleus with a blue lining around it, and the Golgi complex is yellow. Now each of these organelles that I mentioned, nucleus, mitochondrion, Golgi body and endoplasmic reticulum, they have their own membrane. So this cell in some way behaves like a building with multiple rooms. So if this is a building, it has multiple rooms inside it. On the other hand, prokaryote has only one room, it's a one room house. Now cell is the smallest living independent living being, and to enunciate more about the cellular components, it's very important to understand both of them will have cellular membrane, and more often than not a cell wall. Inside they have cytoplasm. Cytoplasm is a water based mixture of macromolecules, biomolecules, which are very important, and this is where life happens, the biochemistry of life happens. Now this green mass both in prokaryote and eukaryote is nuclear material, which is genetic code. This is the code of life, and there are 4 alphabets in code of life: A, T, G, C, and in next lecture we will be talking about A, T, G, C more, but for now it's important to understand that the information stored in this green mass is what tells cell how to deal with the day-to-day activities, the minute-to-minute challenges and issues that come up. These encode how life will work for this cell. In terms of a eukaryote, let's take our example, our green mass, the DNA, will decide what color our skin would be, whether we are going to be humans or not in the first place, how our hair would turn out, and what diseases we are susceptible to. I want to mention that these two pictures on the left for prokaryotic and eukaryotic cell are artistic impressions. These bodies don't really have these colors, but they're helpful to visualize in a class. On the right side we have more real microscopic images of 2 prokaryotes and 1 eukaryote. And the good question that I want you to think about is, why do we have 2 prokaryotic examples and 1 eukaryotic example? We'll talk about it in next lecture, but for now I think it's important to know that eukaryotic and prokaryotic cells are different from each other, and I'll go over these details in detail later. So for now it's just important to know that the eukaryotic cell is much bigger than prokaryotic cell. Eukaryotic cell has multiple organelles with separate individual membranes, whereas prokaryote doesn't. The nuclear material in eukaryote, or genetic material in eukaryote, is much longer and much more complex than one in prokaryote. Eukaryotic nuclear material also have very interesting things like histones, and do not have plasmids that are very often found in prokaryotic cells. Another important difference between prokaryotic and eukaryotic cell is of size. Most prokaryotic cells, here the examples are mycoplasma, gram-positive and gram-negative bacteria, are less than 10 to the power 7 base pair per cell. Most eukaryotes are much larger, and that's the reason why it was so easy and so convenient to sequence the bacteria much before we could manage human genome. In fact, it's interesting to note that in case of prokaryotes, which are bacteria is an example of prokaryote by the way, in case of bacteria, an individual lab could sequence the entire genome, but for human, for many years we had to rely on a massive human genome project. And then the other difference between eukaryote and prokaryote would be that eukaryotic genetic material is linear, whereas for prokaryote it's circular. So if we could unwind this green convoluted mess here for a prokaryotic cell, it will come out to be a perfect circle, and for eukaryotic it will come out to be a straight line. Alrighty, now what does a cell do and what its structure is? Now on left side we have important activities of the cell, such as metabolism, growth and evolution, that it does on its own individual level. On right side we have differentiation, communication, genetic exchange and motility. This is how a cell behaves, interacts with its environment and its colleagues. So let's look at metabolism. Cells take up nutrients, they transform them and expel the waste, much like what higher orders of life do. And in terms of metabolism, it has 2 components, genetic and catalytic. Now genetic activities would include replication, transcription, translation. We'll go over them in next class in much detail. So for now it's just important to know that when it comes to cell activities, it has 2 components, genetic and catalytic, which is basically genetic and chemical. The other important activity for cell is to grow, right? A cell needs to multiply, it needs to grow. So from one cell it needs to gather enough material, enough energy and enough nutrients that it can divide into 2 daughter cells, and so on and so forth. The other activity that cells participate in is evolution. Cells evolve at every step of reproduction. So here we have one cell and it splits into 2 daughter cells. Right here in between this, the replication process, there is a possibility that the daughter cells will be slightly different from the parent cell. And the reason for that is evolution. The genes within the microbes, their genetic material, evolve, change, and as such here we might have one ancestral cell and it will create many progenies, of which the progenies might go and cluster into unique species. And from one ancestral cell we might have very distinct species that look different, behave different and are genetically very diverse. And in fact there is a belief in biological community that we all have one common ancestor, Luca, and we'll talk about it in the next lecture. Then we have on the right we have differentiation, communication, genetic exchange, and motility. And I like to just quickly summarize here that differentiation is when a cell can transform into something else. So we have a living vibrant cell, but conditions around it go really bad and it decides to become a spore. So this is different from growth. Please keep this in mind. Then we have communication. Much like human beings, cells also communicate with each other. They produce these beautiful chemicals which act like messengers, and anyone who catches the chemical and knows how to decode it will know what the message is. And our science suggests that antibiotics are produced by bacteria to talk to each other and to ward off unfamiliar, unfriendly microbes. The other thing cells do is genetic exchange. This is really cool. A cell can share its genetic material with another cell just by touching. This is almost like saying, if I have curly hair and I touch my friend who has straight hair, I can give her genes to have straight hair. Cool, right? And this is also a reason why we have a lot of environmental challenges. So this is very relevant. And next important thing is motility. Cells have capacity, some cells, not all, have capacity to propel them. And in this picture, you have this beautiful microbe with a lot of appendages here. What they do is they twist in a particular direction and they act like a supercoiled spring. So the moment the bacteria decides that, okay, I'm ready to move forward, it will let the spring uncoil and it's propelled forward. When it wants to change direction, it just asks its flagellum to, no, it doesn't ask, it just whips its flagellum in opposite direction and it comes to a standstill, and then it can coil in a different direction to opposite to which it wants to go. So here we have cell structure and activities, a good summary of them, and in the next lecture I will cover more details about cell structure and activities. We will also talk about other applications of applied environmental microbiology, what you are going to learn in this course, and how it will help you become better environmental engineer, environmental scientist, and just improve, and if nothing else, it will definitely improve your awareness and understanding of microbiology around you, help you become better and more productive engineer. So I'll stop this lecture here and continue in the next lecture. Thank you.